Good morning, good afternoon, and good evening to everyone who joins us from around the world. We're so glad you're here. This is Andrew and Renee. Welcome to the Stack series. Today, we are exploring navigating drug toxicity and the AI revolution in drug discovery.
It's quite a massive stack of sources we have today. I mean, an entire library of really cutting-edge research.
It really is. And, our mission in this exploration is to guide you, the listener, through the entire life cycle of pharmacological peril. We're going to examine the very biological mechanisms of how drugs interact with our bodies to cause toxicity.
Right.
And then move into the, frankly, mind-bending challenge of chirality in chemistry. And finally, we'll uncover how artificial intelligence platforms, things like deep talks and agentic AI, are quite literally rewriting the rules of modern medicine.
Yeah, it's a vast landscape we're covering.
Right.
We are essentially mapping out the journey of a molecule from the exact moment it enters your body to the potential havoc it can wreak, and then zooming all the way out to see how supercomputers are trying to predict and prevent that havoc before a drug is even synthesized in the real world.
Which is incredible.
It is. But to understand the future of drug discovery, you really have to understand exactly what goes wrong at a cellular level when things do not go according to plan.
Well, let's start right there. At the cellular level, we have to lay down the biological foundations before we can even talk about the algorithms trying to fix them.
Exactly.
Our primary text here is Casseret and Dole's Toxicology, which is, it's widely considered the absolute foundational text of this field. It outlines 4 distinct steps of toxicity, and step 1 is delivery, from the site of exposure to the target.
Right, the delivery.
For you listening, the best way to visualize this is to think of a drug molecule like a commuter trying to get to work. But this isn't a normal commute. This is a commute through a massive, chaotic, actively hostile city that basically wants to dismantle your car.
The commute is actually the perfect way to frame this. The journey begins with absorption. For any drug to have a systemic effect, whether that's therapeutic or toxic, it first has to get on the main highway. It needs to enter the systemic blood circulation.
Right.
But before our chemical commuter even reaches the on-ramp, it faces some really severe hurdles. We call this pre-systemic elimination. So if you swallow a pill, it has to survive the highly acidic environment of your stomach.
Which is basically a vat of acid.
Exactly. Then it gets absorbed through the intestines and routed directly to the liver. And the liver is the body's primary chemical checkpoint. It is packed with metabolizing enzymes whose sole job is to identify foreign substances and just destroy them or filter them out.
So the commuter might get pulled over and their car impounded before they ever reach the highway.
Precisely. And assuming our molecule survives the liver's checkpoint and actually makes it onto the highway of the bloodstream, the journey is still far from over. Now we enter the distribution phase.
Because the drug is circulating, but it needs to get off at a very specific exit to reach its target tissue.
Right. And the body has physical barriers. Things like lipid bilayers surrounding every single cell, acting like toll booths.
Which brings up a really critical chemical property, lipophilicity. This simply means how well a molecule dissolves in fats or lipids.
Yeah, a highly lipophilic drug basically has a VIP pass. Because cell membranes are made of lipids, a fat-soluble molecule can slip right through the toll booth without needing a specialized transport channel. It distributes rapidly and widely.
But the body doesn't just let that happen, right? It mounts a defense against this distribution.
It absolutely does. You have specialized transporter proteins acting as bouncers, actively pumping foreign substances back out of cells. You also have proteins in the blood, like albumin, that can actually bind to the drug.
And if the drug is tightly bound to a blood protein,
then it's trapped in the vehicle. It cannot interact with its target receptor at all.
It sounds like a constant biological tug of war. The drug is trying to distribute and the body is trying to trap it or pump it away. And eventually, the body's ultimate goal is to kick the commuter out of the city entirely.
Yes, which brings us to excretion versus reabsorption. The blood eventually filters through the kidneys. The kidneys act as a massive sorting facility, dumping waste into the urine. But depending on the chemical properties of our drug, it might accidentally get reabsorbed from the kidneys back into the bloodstream instead of leaving the body.
And reabsorption is a massive variable in toxicity, isn't it?
Oh, massive. If the drug keeps getting recycled into the blood, the exposure time is prolonged and the concentration just builds and builds. But perhaps the most counterintuitive part of this entire delivery step is the concept of toxication versus detoxication.
Yeah, let's break that down because we usually assume the liver breaks down a drug to make it harmless. It's detoxication, right?
Right. The liver attaches a bulky, water-soluble molecule to the drug, so the kidneys can easily excrete it. However, sometimes those exact same liver enzymes, in their attempt to break down a molecule, inadvertently convert a perfectly safe parent drug into a highly reactive, incredibly dangerous intermediate form.
Wow.
That is toxication. The body accidentally manufactures what toxicologists call the ultimate toxicant.
I want to pause on that term, the ultimate toxicant, because it sets up the next phase perfectly. Our commuter has arrived at their destination, but because of the liver's meddling, they've turned into an absolute menace.
They're heavily armed now.
Right. This is step 2 in Kayser and Dole's framework, reaction of the ultimate toxicant with the target molecule. So the drug is now interacting directly with the body's proteins, lipids, or DNA. How exactly does this chemical vandalism happen?
The vandalism happens through a few highly specific chemical reactions. The most common is non-covalent binding. For you listening, think of this like Velcro. The toxicant sticks to a receptor or an enzyme, physically blocking its function.
But because it's just Velcro, it can eventually unstick. It's reversible.
Exactly. It's an annoyance, but it's reversible. The much more dangerous interaction is covalent binding. This is not Velcro. This is industrial superglue. The toxicant forms a permanent electron bond with a critical protein or a strand of DNA, fundamentally and irreversibly altering its physical structure.
Supergluing a foreign chemical to your DNA sounds like a guaranteed disaster. Does the body have any way to rip that superglue off?
Usually, no. The protein or the DNA segment is permanently ruined and has to be entirely destroyed and rebuilt by the cell. But covalent binding isn't the only aggressive reaction. You also have hydrogen abstraction.
What does that look like?
This is where the toxic molecule literally rips a hydrogen atom off an endogenous, healthy molecule.
Yeah.
Imagine driving down the highway and someone just rips a wheel off your car.
That'll cause a crash.
It causes a huge crash. The molecule that lost its hydrogen becomes a highly destructive free radical, which goes on to damage other molecules in a massive chain reaction. Then you have electron transfer, where the toxicant steals an electron from or forces an extra electron onto another molecule.
Kind of passing a chemical hot potato.
Exactly, passing a hot potato that leads to severe oxidative stress. And finally, there are enzymatic reactions, where the toxicant acts like a rogue factory worker, catalyzing chemical reactions that shouldn't be happening at all.
So the toxicant is supergluing things together, ripping wheels off cars, and passing around explosive hot potatoes. What is the actual outcome for the target molecule? I mean, what happens to the poor protein that just got attacked?
There are three primary outcomes at the molecular level. First is dysfunction. The target protein just stops working. If it's an enzyme responsible for creating energy, energy production stops.
Makes sense.
Second is destruction. The toxicant physically breaks the target molecule apart. And the third outcome is often the most insidious, neoantigen formation.
Let's break down neoantigen for the listener. Neo meaning new, and antigen meaning something that triggers the immune system.
Exactly. When a toxicant covalently binds, superglues itself to a normal protein in your body, it changes the shape of that protein. It essentially puts a disguise on it. Your own immune system constantly patrols the body, looking for foreign shapes.
And it spots this disguise.
Yes. It sees this altered protein, fails to recognize it as self, and mounts a full-scale attack. Your immune system starts destroying your own tissues because the drug tricked it.
Which is terrifying. And this cascading failure brings us to step 3, cellular dysfunction. The toxicant has wreaked havoc on individual molecules, and now the entire cell is starting to realize it's under attack. The local vandalism is basically turning into a citywide blackout.
That's a great way to put it. The toxicant-induced cellular dysregulation impacts the cell on two main fronts. First, it causes dysregulation of gene expression. Every cell relies on its genetic blueprint to know which proteins to build and when to build them.
Right, the instruction manual.
Right. If the toxicant interferes with the transcription factors reading that blueprint, the cell gets deeply confused. It might stop producing essential proteins, or it might start wildly overproducing them. It's like a corrupted hard drive feeding bad instructions to a factory.
And the second front.
Second, you have the dysregulation of ongoing cellular activity. The minute-to-minute operations firing electrical signals, secreting hormones, they just grind to a halt.
But a cell is a living thing. If it can't function, it has to try to keep the lights on somehow, right? What happens to its internal maintenance?
That is where the real crisis hits. We see the impairment of internal cellular maintenance. A cell spends a massive amount of energy just keeping itself alive, pumping calcium out, synthesizing ATP for energy. When a toxicant impairs these basic survival mechanisms, the cell essentially runs out of power.
And what happens when the power goes out completely?
The calcium levels inside the cell spike uncontrollably, which acts as a self-destruct signal. It triggers toxic cell death. And we can't forget the impairment of external cellular maintenance.
Because cells don't exist in a vacuum.
No, they maintain their surrounding environment. For example, liver cells produce factors that allow your blood to clot. If the toxicant disrupts this external maintenance, the local cellular damage ripples out to cause systemic failure in the entire human body.
Which means the body has to respond. It can't just let the cells die and the tissues melt. And this is the final step, step 4, inappropriate repair and adaptation. The body sends in the emergency crews.
The body does have sophisticated levels of defense. At the lowest level, there is molecular repair. Specialized enzymes act like molecular mechanics. They scan the DNA for superglue toxicants or broken strands, they cut the damage out, and they patch it.
That's incredible.
It is. Then there is cellular repair, where the cell breaks down its own damaged organelles and recycles the parts. And finally, tissue repair, where the immune system clears out dead cells and stimulates neighboring healthy cells to divide and fill the gap.
Does the body try to adapt to the drug itself?
It does. The body tries to adapt to the assault. It might decrease delivery by building more of those efflux pumps to throw the drug out. Or it might decrease target density, literally pulling receptors inside the cell so the drug has nothing to bind to.
It's an incredibly resilient system. But the title of this step is inappropriate repair. So what happens when the emergency crews fail, or worse, when they just overreact?
That is when you transition from microscopic damage to visible clinical toxicity in the patient. If the damage is too severe and repair completely fails, you get tissue necrosis.
Which is?
This is uncontrolled, messy cell death. The cells literally burst open, spilling their contents and causing massive, painful inflammation in the surrounding tissue.
And if the repair process works but goes overboard?
Then you get fibrosis. The body lays down too much structural collagen, resulting in excessive scar tissue that hardens organs like the liver or lungs and destroys their flexibility and overall function.
And the most severe outcome?
Carcinogenesis. This happens when molecular repair fails to fix a damaged strand of DNA, but the cell doesn't actually die. Instead, the cell survives with a mutated genetic code and begins to divide uncontrollably. The toxicant has initiated cancer.
So that is the fundamental biology of how damage happens, but let's look at the why. When a patient actually takes a pill, why does it trigger this cascade in some people but not in others? How do we categorize these adverse effects in the real world?
The first and most critical category is patient factors. Toxicity is not just a property of the drug, it is a dynamic interaction with the host. A drug that is safe for a healthy 30-year-old might be lethal to an 80-year-old.
Because their biology is just different.
Exactly. Age, genetics, and comorbidities drastically alter both pharmacokinetics, which is how the body moves the drug, and pharmacodynamics, which is what the drug does to the body. So if a patient has even mild kidney dysfunction, they cannot excrete the drug at a normal rate. The commuter gets stuck in the city. A standard safe dose suddenly accumulates into a massive toxic overdose simply because the patient's biological filtration system was sluggish.
Let's talk about the drug's intended mechanism, because sometimes the drug does exactly what it's supposed to do, and that is the actual problem. We call these on-target adverse effects. And the analogy I find helpful for you listening is a lock and key. The drug is the key, the specific cellular receptor is the lock. But sometimes the key turns the lock so hard or opens so many doors that it breaks the system.
That captures it perfectly. The drug hits the exact correct target, but to an excessive degree. Take diphenhydramine, a very common over-the-counter allergy medication. Its target is the histamine receptor to stop allergic inflammation.
But it makes you sleepy.
Right, because the histamine receptors also exist in the central nervous system to regulate wakefulness. The drug hits the intended target, but because it crosses into the brain, it causes profound sedation.
Are there other examples?
A classic example is statins, prescribed to tens of millions of people to lower cholesterol. They work by inhibiting an enzyme called HMG-CoA reductase in the liver. But that same enzyme exists in muscle tissue. If the statin inhibits the enzyme too heavily in the muscles, it causes severe muscle toxicity and breakdown. The drug isn't failing, it's succeeding too aggressively.
But then you have the chemical accidents, off-target adverse effects. The key gets shoved into the completely wrong lock because the locks happen to look similar in the dark.
These are the structural accidents that keep medicinal chemists awake at night. The most infamous example is terfenadine. It was a blockbuster antihistamine, widely prescribed because it didn't cause the sedation of older drugs. It was brilliant.
Until it wasn't.
Right. It had a hidden lethal off-target effect.
Right.
The physical structure of terfenadine inadvertently allowed it to bind to and inhibit cardiac potassium channels, specifically the HERG channel in the heart.
We're going to explore HERG much deeper later because it is a massive focal point for AI. But for now, what happens when you inhibit that specific channel?
The HERG channel is responsible for resetting the heart's electrical system after every single beat. When terfenadine blocked it, the heart's electrical rhythm descended into complete chaos, causing fatal arrhythmias. Patients were dying simply because a drug designed to stop a runny nose accidentally fit into a cardiac electrical switch. It had to be withdrawn from the market entirely.
And as we mentioned during the delivery step, sometimes the parent drug isn't the problem at all. Sometimes the liver manufactures the poison. This is the production of toxic metabolites, and the most common example of this is sitting in almost everyone's medicine cabinet right now, acetaminophen or APAP-P.
Acetaminophen is an incredibly safe drug at normal therapeutic doses. But the way the liver metabolizes it is a masterclass in the delicate balance of toxicity. When the liver breaks down APAP, a small percentage of it is converted into a highly reactive intermediate molecule, specifically a quinone species.
The ultimate toxicant again.
Yes, this quinone is a classic ultimate toxicant. It desperately wants to covalently bind to liver proteins and destroy them.
So why doesn't Tylenol destroy our livers every time we take it?
Because the body has a dedicated defense mechanism. The liver maintains a large stockpile of a molecule called glutathione. Glutathione acts as a chemical sponge. It intercepts the toxic quinone species and neutralizes it before it can damage the liver proteins.
But if you take too much...
If you take a massive overdose of APAP, the liver produces so much of the toxic quinone that it completely depletes the glutathione stockpile. Once the sponge is gone, the quinone runs rampant, causing massive oxidative stress and widespread tissue necrosis.
The liver just fails.
The liver rapidly destroys itself, leading to fatal acute liver failure. The APAP itself wasn't directly toxic. The liver simply ran out of the resources needed to clean up its own metabolic waste.
It really illustrates how precarious our internal chemistry is. I want to touch on harmful immune responses too, because you brought up neoantigens earlier.
Right.
We can broadly divide these into two types, right?
Broadly, yes. Type I reactions are what most people think of as a drug allergy. These are immediate, severe, and mediated by IgE antibodies. You take a medication, and within minutes, your body triggers mast cells to release massive amounts of histamine. You get hives, airway constriction, and potentially fatal anaphylaxis. It is a rapid-fire immune explosion.
And the second?
FIV reactions. These are T-cell mediated and delayed. You might take a drug every day for 3 weeks with absolutely no issue. But slowly, behind the scenes, your T cells are learning to recognize the drug protein complexes as foreign. When they finally mount an attack, it results in severe delayed skin reactions or systemic organ inflammation.
And that leaves us with the final category, which frankly sounds like a physician's worst nightmare: idiosyncratic toxicity.
Idiosyncratic simply means peculiar to the individual. These reactions are terrifying because they are entirely unpredictable. They do not follow normal dose-response curves. You could give a patient a microdose, and they might experience catastrophic liver failure. You could give them a massive dose, and they might be totally fine.
Why does that happen?
These reactions occur due to incredibly rare, patient-specific genetic mutations or unique immune system quirks. Because they might only happen in 1 out of every 50,000 people, they are nearly impossible to detect during standard clinical trials. You only discover them after the drug has been approved and prescribed to millions.
So when pharmaceutical companies are trying to navigate this minefield, they rely heavily on measuring specific physicochemical properties. They use a framework called ADMET.
ADMET is the fundamental language of medicinal chemistry. It stands for absorption, distribution, metabolism, excretion, and toxicity. Every single molecular tweak a chemist makes is designed to optimize these 5 variables.
We discussed lipophilicity earlier, fat solubility.
Exactly. High lipophilicity is great for absorption and distribution.
Yeah.
Gets the drug into the brain or the tumor, but it's terrible for excretion and toxicity because fat-soluble drugs linger in adipose tissue, building up over time and causing long-term damage.
So it's a balancing act.
It's entirely a balancing act. Much of a medicinal chemist's job involves lipophilicity-reducing transformations. They add slightly more water-soluble molecular groups to a drug prototype, trying to walk the razor's edge. making it just lipophilic enough to reach the target, but water-soluble enough that the kidneys can flush it out before it causes a disaster.
That perfectly encapsulates what we think we know. We understand the biology, the mechanisms, and the categories of failure, but science is never settled. If the body is this much of an unpredictable minefield, how do we stop sending chemical commuters in blind? This transitions us into the active debates in the field where the scientific community is currently clashing over the best way forward. First up is the ethical and scientific push for the 3 Rs and the future of animal testing.
The 3 Rs represent a massive paradigm shift. The principle stands for replacement, reduction, and refinement of animal research and toxicology. Historically, the only way to know if a drug would destroy a liver was to give it to an animal and check.
Which is ethically fraught.
Immensely. Ethically, there is immense societal pressure to end this. Scientifically, there is a growing realization that animal models often fail to accurately predict human toxicity anyway because a mouse is not a human. The goal is to replace animals with computational models, reduce the number of animals required when testing is unavoidable, and refine the procedures to minimize any suffering.
And the debate really centers around the replacement aspects, specifically the use of structural alerts in computer models. A structural alert is essentially a software red flag. A computer scans a newly designed molecule and says, hey, this specific arrangement of carbon and nitrogen looks a lot like terfenadine. It might block the HERG channel. Do not synthesize this.
In theory, structural alerts are the perfect solution. They allow us to predict toxicity in a virtual environment without harming a single living creature. But the active debate within the scientific community is fierce because these computational flags are currently incredibly flawed. Well, a computer might flag a molecule as highly toxic based purely on a 2D drawing of its structure. But in the real, highly complex 3D environment of the human body, that molecule might fold in a way that makes it completely safe.
Oh, I see.
Conversely, and much more dangerously, the algorithm might look at a novel chemical structure, fail to recognize any known toxic patterns, and declare it perfectly safe, completely missing a new mechanism of toxicity.
So we are caught between a rock and a hard place. We want to stop animal testing, but we can't fully trust the algorithms yet.
Precisely. The current, albeit tense consensus, is that computational models must be integrated with traditional experimental methods. The software can filter out the obvious poisons, significantly reducing the animal testing required, but it cannot yet replace the final biological proof of safety.
Which leads us directly into the biggest, most glaring flaw in those algorithms right now, a concept that is absolutely mind-bending, the chiral blind spot in AI and pharmacology. To understand why the algorithms are failing, we have to talk about chirality. What on earth is it?
To understand chirality, we have to look at stereochemistry and isomers. I'll use the classic and honestly most effective analogy: your left and right hands. For those of you listening, hold your hands out in front of you. They are composed of the exact same elements: 4 fingers, 1 thumb, a palm. They are mirror images of each other.
Right.
But if you try to place your left hand perfectly on top of your right hand with both palms facing down, the thumbs point in opposite directions. They are non-superimposable.
They look identical on a flat piece of paper, but in 3D space, they are fundamentally different shapes.
And that 3D shape dictates function. A left hand will simply not fit into a right-handed glove. Molecules behave the exact same way. Many complex drug molecules possess a handedness. They exist as enantiomers, which are mirror image isomers of the exact same chemical formula. One version of the molecule is the left hand, and the other version is the right hand. Now remember that the biological receptors in your body, the locks, are highly complex 3D structures. They're gloves.
So the left-handed molecule might slide perfectly into the receptor, turning the lock and curing the disease. But the right-handed molecule...
The right-handed molecule might bounce harmlessly off the receptor because it doesn't fit the glove. Or, far more catastrophically, the right-handed molecule might fit perfectly into a completely different receptor somewhere else in the body, triggering a severe toxic cascade. Biology speaks strictly in 3D.
And here is where the active debate and the crisis in AI overlap. Current algorithms and AI models are described as being stereosloppy.
Stereosloppy is the critical term. Most current machine learning models used to predict drug toxicity read molecules in a 2D format. They ingest a string of text that tells them how many carbon and oxygen atoms are present and exactly how they are connected. But they are completely blind to the 3D twist. They lack the spatial awareness to differentiate between the left hand and the right hand.
So to a stereosloppy AI, both enantiomers look absolutely identical.
Yes, this completely breaks the drug pipeline. Because an AI might confidently predict that a molecule is safe, and highly efficacious, entirely unaware that its mirror image is a lethal poison.
But surely the human regulators know about this, right? It's not just an AI problem, it's a regulatory blind spot, too.
It is a massive regulatory blind spot. Historically, many drug labels simply miss the twist. For decades, pharmaceutical companies sold drugs as racemates. A racemate is a 50-50 mixture of both the left-handed and right-handed molecules.
Why would they do that?
They did this because chemically separating the two is incredibly difficult and expensive. They essentially hoped the wrong hand would just be inactive baggage. Furthermore, there is hidden chirality in drug metabolites. You might design a perfectly safe non-chiral parent drug, but when the liver enzymes break it down, they might inadvertently twist the molecule, creating a chiral metabolite. The AI models completely fail to predict this metabolic blind spot.
So, if biology speaks in 3D and our computers only read in 2D, what is the call to action? How do we fix this?
The urgent call to action is that artificial intelligence must develop chiral intelligence. We need AI models possessing true chiral literacy, the ability to model, simulate, and predict molecular interactions in full dynamic 3D space. We can no longer rely on stereoscopy models to design the medicines of the future.
We'd have to force the issue.
We must mandate safer, smarter, and antioselective medicines. This requires a massive investment in asymmetric synthesis, which is the chemical technology required to manufacture only the exact, correct handed molecule we want, with zero contamination from its mirror image. If our AI cannot see in 3D, it cannot guide asymmetric synthesis.
That realization is the perfect bridge into our next major area. We are looking at the frontiers, what might change everything, because despite the chiral blind spot, the sheer scale of the AI revolution in toxicity prediction is staggering. Let's look at the foundational effort to fix these predictive models, the Tox 21 10K challenge. What was the scale of this initiative?
The Tox 21 10K challenge was a monumental collaborative initiative spearheaded by the NIH and the EPA. The regulatory agencies recognized that testing hundreds of thousands of environmental chemicals and drug candidates on animals was mathematically impossible. So they compiled an enormous database of 10,000 distinct chemical profiles.
And what did they do with it?
They provided this vast data set to computational researchers worldwide and essentially issued a challenge. Use your algorithms to model these 10,000 chemicals, tell us which ones will be toxic in a living organism, and more importantly, predict the exact biological mechanism of that toxicity.
And out of all the models submitted globally, one absolutely dominated the field. It was an AI called DeepTox. How did it work, and what specifically did it excel at predicting?
DeepTox was a true revelation for the field. It utilized deep learning, which involves highly layered artificial neural networks that can identify nonlinear patterns in massive datasets that human engineers would honestly never spot. The Tox 21 challenge was divided into sub-challenges based on specific biological pathways. DeepTox proved incredibly adept at predicting toxicity across nuclear receptors.
Let's break down why nuclear receptors are so important. These are proteins inside the cell that directly interact with DNA, correct?
Correct. They govern fundamental biological processes. DeepTox was highly accurate at predicting disruption of the AISHR, the aryl hydrocarbon receptor. This is the primary receptor that mediates the severe toxicity of environmental pollutants like dioxins.
Did it model others?
It also accurately modeled the AR, the androgen receptor, and the ER, the estrogen receptor. Predicting disruption here is crucial for avoiding endocrine-disrupting chemicals that cause reproductive toxicity. And finally, it excelled at predicting interactions with PPAR gamma, a receptor deeply involved in fatty acid storage and glucose metabolism.
So DeepTox proved that deep learning could look at a 2D chemical structure and highly accurately predict if it would catastrophically disrupt these crucial hormonal pathways.
Exactly.
And it didn't just stop at hormonal disruption. DeepTox also crushed the predictions for stress response panels.
It did. It accurately predicted interactions with pathways like P53. For context, the P53 protein is famously known as the guardian of the genome. It is a critical tumor suppressor. If a chemical disrupts P53, the risk of carcinogenesis skyrockets. DeepTox also modeled the HSE, the heat shock response element, which cells use to survive extreme protein-damaging stress. By predicting these stress responses, DeepTox proved that AI could anticipate the exact cellular dysfunctions we discussed in Step 3 of the biological foundations.
That challenge proved the concept, and now we are seeing incredible platform innovations building on that success. We have integrated systems now, like ADMET Lab 2.0.
ADMET Lab 2.0 is the democratization of this technology. It is a comprehensive online platform that allows medicinal chemists anywhere in the world to upload a molecular structure and receive rapid, highly accurate predictions of its full ADMET profile. It integrates dozens of predictive models into one single interface. And alongside platforms predicting chemical properties, we have to acknowledge the revolution in structural biology, primarily AlphaFold 2.
AlphaFold 2, developed by DeepMind. I know it isn't specifically a toxicity predictor, but its impact on this field is massive.
It is arguably the most significant biological breakthrough of the decade. As showcased in the CASP-14 assessment, AlphaFold 2 essentially solved the protein folding problem. It can predict the complex 3D structure of a protein purely from its 1D amino acid sequence.
And why does this matter for toxicity?
Because if we know the exact 3D structure of every off-target receptor in the human body, we can simulate whether our drug will accidentally bind to them. It is the key to solving the off-target adverse effects we talked about earlier.
Which brings me right back to the most terrifying off-target effect of all, terfenadine and the HERG potassium channel. Blocking HERG causes fatal heart arrhythmias. Because of terfenadine, predicting HERG blockage is the holy grail of early drug discovery. No one wants to accidentally design a drug that stops the heart. How is modern AI tackling the HERG problem specifically?
The HERG channel is notoriously difficult to model because it has a very accommodating internal cavity. It accidentally binds to a vast array of totally different molecular structures. Because it is so complex, AI researchers cannot rely on a single algorithm. They use highly specific ensemble approaches.
An ensemble approach.
It means you train a diverse group of different machine learning models and combine their outputs. For HERG cardiotoxicity prediction, the workhorse of this ensemble is the random forest model, often abbreviated as the RP model.
Okay, I want to take a moment to really visualize this for you, the listener. What exactly is a random forest model in the context of predicting if a drug will stop your heart?
Imagine you have a highly complex life or death decision to make regarding a new drug. You wouldn't just ask one expert, you would ask 100 different experts, all with slightly different backgrounds. A random forest model algorithmically builds a literal forest of individual decision trees. Each individual tree is fed slightly different data about the molecule and asks a series of yes or no questions. Does it have a nitrogen ring? Is it highly lipophilic?
And then what?
Each tree makes a final vote, toxic or safe. The forest then tallies all the votes and outputs the majority decision.
But how does the tree actually look at the molecule to ask those questions? They don't have eyes.
They look at the molecule using something called ECFP8 fingerprint sets.
Fingerprints for molecules.
Yes, extended connectivity fingerprints. You can think of it as a mathematical map of an atom's neighborhood. The algorithm selects one atom in the molecule, say a specific carbon. It then maps out every other atom and bond connected to it, radiating outward in a circle up to a maximum diameter of 8 bonds. That's the 8 in ECFP8. It does this for every single atom, compiling a massive list of all the structural neighborhoods present in the drug. It translates the physical 3D molecule into a long string of binary ones and zeros, a unique mathematical fingerprint that the random forest can easily read and compare against known HRG blockers.
That is fascinating. So the fingerprint gives the trees the data. But I noticed in the research that the data scientists spent a lot of time tweaking the tree depths, testing models with a depth of 2 all the way up to a depth of 10. Why is the depth of the tree so crucial to get right?
Tree depth is simply how many sequential questions a single decision tree is allowed to ask before it is forced to cast its vote. If you restrict the tree to a depth of 2, it can only ask 2 broad questions. Is it big? Is it fat-soluble? If you allow a depth of 10, it can ask a highly granular, complex sequence of 10 conditional questions.
So they test depths from 2 up to 10 to find a balance.
Yes, they want to find the mathematical sweet spot that balances 4 critical classification metrics: true positives, false negatives, sensitivity, and specificity.
Let's define those in terms of a patient taking a pill. A true positive, or TP, is when the model correctly flags a dangerous HERG blocker. That saves lives. A false negative, or FN, is the nightmare scenario. The model looks at a lethal heart-stopping drug and confidently tells the chemist, this is perfectly safe.
Exactly. And those outcomes dictate the model's overall performance metrics. Sensitivity, abbreviated as SE, measures the proportion of actual toxic compounds that the model correctly caught. Specificity, or SP, measures the proportion of safe compounds it correctly identified as safe.
And here is why tree depth matters.
Right. If a tree is too shallow, like depth 2, it is overly simplistic. It might have high specificity. It rarely falsely accuses a safe drug, but it'll have terrible sensitivity. It misses the nuanced structural traps and produces massive amounts of dangerous false negatives.
So why not just make the tree depth 1,000? Let it ask infinite questions.
Because if the tree is too deep, like depth 10 or beyond, you run into the problem of overfitting. The model stops learning general rules and starts memorizing the exact training data it was given. It will perfectly identify the old toxic drugs, but when you show it a completely brand new, unseen chemical structure, it panics and fails to generalize. It starts flagging perfectly safe drugs as toxic, ruining viable drug candidates. Finding the optimal tree depth is the ultimate high-stakes balancing act of data science, minimizing false negatives while preserving the pipeline.
It is incredible how much mathematics is required just to ensure a pill doesn't disrupt an electrical channel in the heart. But the AI revolution isn't stopping at better prediction algorithms. We're crossing a threshold into a completely new era. The literature calls it the Science Dawn, spelled SCAINCE. This is the dawn of agentic AI and autonomous labs.
This represents a fundamental architectural shift in how human beings conduct scientific research. We have to clearly define the agentic divide. For the past 10 years, we have utilized AI agents. An AI agent is a highly advanced form of automation. You, the human scientist, act as the commander. You give the AI agent a highly specific, bounded task, like run a random forest model with a tree depth of 6 on this library of 1,000 molecules using ECFP8 fingerprints. The AI executes the task brilliantly and hands you the results. It is an assistant.
But agentic AI operates on the other side of that divide. It is not an assistant.
No, agentic AI possesses what we call emergent system-level intelligence. It does not wait for a step-by-step task list. You give it a high-level goal, and it plans, reasons, and adapts autonomously. You tell the agentic AI, invent a molecule that cures this specific cancer without blocking the hERG channel.
And it just does it.
The AI will autonomously design a molecule, write the chemical synthesis instructions, transmit those instructions to a fully automated robotic laboratory, wait for the physical robots to synthesize and test the drug on biological cells, interpret the toxicity data from those assays, realize its initial design was slightly too lipophilic, autonomously redesign the molecule to be safer, and tell the robots to build the new version. It iterates entirely on its own.
That is staggering. The AI is no longer just processing data. The entire physical structure of the scientific method is being redesigned to serve intelligent machines. They are the principal investigators now.
And the efficiency metrics we are already seeing are hard to fathom. We have platforms operating right now, like Eureka LS, that function as autonomous AI agent squads for drug discovery. By completely automating the tedious biopharma busywork from drafting complex 3D chemical structures to cleaning and interpreting vast datasets of assay results, platforms like Eureka LS are currently saving 50-plus research teams over 300 hours a month. That is not just a marginal improvement. That is an exponential acceleration of the pace of human discovery.
And what exactly are these supercomputers and brilliant chemists discovering? Let's move to a biological frontier that completely flips the script on how we treat disease. We are moving away from traditional pharmacology and entering the era of proteolytic rewriting and targeted protein degradation.
Targeted protein degradation, or TPD, is perhaps the most profound conceptual shift in pharmacology in a century. For decades, the entire pharmaceutical industry operated on one basic premise: inhibition. You find a pathogenic, disease-causing protein, and you design a small molecule to bind to its active site and block it from working. The lock and key analogy. You plug the keyhole so the bad protein can't function.
But TPD says, why just block the keyhole when we can blow up the entire door?
Precisely. TPD shifts from merely inhibiting a protein to actively erasing it from existence. It utilizes the cell's own internal waste disposal machinery. The drugs used in TPD are called degraders. They're essentially molecular matchmakers. One end of the drug binds to the disease-causing protein, and the other end binds to an enzyme in the cell whose job is to tag garbage for destruction.
So it forces a connection.
Right. The drug physically drags the bad protein to the cell's trash compactor, where it is shredded into base amino acids.
I want to look at a specific frontier example of this because it sounds like science fiction, the concept of spatial collapse.
Spatial collapse is a brilliant application of TPD. Let's look at carriase. Carriase is a notorious oncogene. Mutated carriase proteins drive a massive percentage of deadly human cancers. For over 40 years, carriase was labeled undruggable because its physical structure is perfectly smooth. It doesn't have a deep active site, a keyhole for a traditional drug to plug. It just bounced every drug away.
Undruggable until now.
Exactly.
Yeah.
Because for carriase to cause cancer, it must be physically transported from the center of the cell out to the cell membrane. It requires a shuttle protein called PDE to carry it there.
So if you can't attack carry S directly because it has no keyhole, you attack its transportation.
Yes. Researchers developed a pharmacologic degradation strategy, specifically targeting the PDE shuttle. By using a degrader drug to actively erase the PDE proteins, they disrupted the membrane localization of KRAS. The KRAS proteins are still sitting there, fully mutated and ready to cause cancer, but without their shuttle, they are stranded in the center of the cell. They never reach the membrane, and the entire oncogenic signaling pathway completely collapses. It is spatial pharmacology. We cured the problem not by inhibiting the enzyme, but by physically moving it to the wrong location.
That is a masterstroke of biological engineering, and there's another powerful example of TPD in the research: precision myeloma therapy.
Yes, the clinical utility of IKZF1 and IKZF3 degradation. Multiple myeloma is a devastating cancer of the plasma cells in the bone marrow. IKZF1 and IKZF3 are transcription factors, proteins that control gene expression, that are absolutely critical for the survival of these malignant plasma cells. Imagine the cancer cells as heavily fortified castles, and the IKZF proteins are the commanders keeping the defenses organized.
So a traditional drug might try to block the commander's radios, but a TPD drug.
A TPD drug sneaks into the castle, tags the commanders as garbage, and the cell's own machinery assassinates them. By designing specific degrader drugs that erase IKZF1 and IKZF3, the malignant plasma cells literally forget how to survive and undergo rapid cell death.
But there is a dual action here.
There is. Erasing these specific proteins simultaneously reprograms the surrounding immune microenvironment. It alters the chemical signals the tumor sends out, effectively opening the gates of the castle and making the tumor highly visible and vulnerable to the patient's own immune system. This dual action, erasing the cancer's survival commanders while recruiting the immune system, is entirely redefining the standard of therapy for both refractory, hard-to-treat cases and frontline multiple myeloma.
It's an incredibly elegant, two-pronged attack, and the innovation isn't just happening in protein degradation. Let's briefly touch on a few other cutting-edge modalities. What is lipid alchemy?
Lipid alchemy refers to fascinating new discoveries leveraging complex natural products, specifically a class of molecules called ganodermal lanostains. Researchers are using these compounds to fundamentally rewire hepatocyte, or liver cell fat chemistry. These lanostains act as sterile mimetic natural products. That means their physical 3D structure closely mimics cholesterol.
So they trick the liver.
They trick the liver's internal sensors. By mimicking sterols, they activate a critical master switch pathway called AMPK. Activating AMPK forcibly regulates and shuts down the biosynthesis of excess fatty acids and cholesterol in the liver. It offers a completely novel, natural product-derived mechanism for treating metabolic diseases, sidestepping the severe on-target muscle toxicity we often see with traditional statins.
We are hacking the liver's metabolism with mushroom derivatives. And in the realm of immunology, we have breakthroughs with STING nanoplatforms. You have to explain STING because it sounds aggressive.
STING stands for Stimulator of Interferon Genes. It is an incredibly powerful ancient signaling pathway that triggers the body's innate immune system. If you artificially activate the STING pathway, the body mounts a massive, aggressive immune assault. In theory, this is the perfect way to destroy a solid tumor. The problem is toxicity. If you inject a STING activator into a patient's bloodstream, it activates immunity systemically. It causes a body-wide, uncontrolled inflammatory storm that can be immediately lethal.
So how do you activate it without killing the patient?
You use stimuli-responsive STING nanoplatforms. These are engineered nanoparticles that act like microscopic smart bombs. They encapsulate the STING-activating drug, hiding it from the body. These nanoplatforms are chemically designed to only break open and release their payload when they encounter the specific, highly acidic, and enzyme-rich microenvironment found exclusively inside a solid tumor.
That is brilliant.
They circulate harmlessly through the blood. But the moment they enter the tumor, they detonate. They activate the massive innate immune response locally, turning the tumor into a war zone while completely sparing the rest of the body from systemic toxicity.
A smart bomb that only arms itself when it recognizes the exact chemical signature of the enemy bunker. Finally, let's talk about viral immunology, the enduring blockade discoveries regarding noroviruses.
This is a discovery that challenges our fundamental understanding of how long immune protection can last. Noroviruses are highly mutable, rapidly evolving viruses that cause severe gastroenteritis. Historically, we believed immunity to them was fleeting. But researchers recently discovered 5-year functional antibody persistence against emerging norovirus strains, specifically the GII.4 and GII.1c variants, following a natural infection.
5 years of active blockade against a rapidly mutating virus.
Yes. The human body is somehow generating and maintaining a highly durable functional antibody blockade against these specific viral structures for half a decade. If we can reverse engineer how the immune system achieves this specific enduring blockade, we could fundamentally revolutionize how we design vaccines, not just for norovirus, but for all highly mutable viruses.
Okay, we've covered the microscopic biology, the computational models, and the futuristic frontiers of protein degradation and smart bomb nanoparticles. But it is time to zoom all the way out. This brings us to the implications. What does all of this mean for the real world, for ethics, for economics, and for human society? Because engineering a miracle in a highly controlled laboratory is completely different from delivering it to millions of patients safely. Let's start with clinical and economics.
The economics of drug discovery are undergoing a forced, rapid evolution. We are moving away from a very siloed, linear model, a shift heavily championed by thought leaders like Dr. Mark Nelson at Numetics. The old model was highly inefficient. Biologists would identify a disease target, medicinal chemists would spend years designing a highly complex molecule to hit it, and only then would they hand it over to the manufacturing engineers and say, figure out how to make a ton of this.
And I imagine the engineers often realized the molecule was physically impossible to mass produce affordably.
Exactly. You end up with a miracle cure that costs $10 million a dose. The new paradigm integrates medicinal chemistry with scalable API synthesis, active pharmaceutical ingredient synthesis for the very earliest computational design stages. You do not finalize a molecular design unless the AI has simultaneously designed a cost-effective, scalable chemical pathway to manufacture it by the ton. Economic viability is now treated as a fundamental physicochemical property of the drug itself.
It is about engineering for reality, not just the petri dish. And there is a specific methodology for handling this complexity during development, utilized at Exelixis, called the Kirkovsky playbook.
Dr. Lear Kirkovsky's approach is foundational for modern oncology. The Kirkovsky playbook operates on the principle that mastering ADME, absorption, distribution, metabolism, excretion, is not just about safety, it is the central decision engine that dictates whether a complex drug program lives or dies. This is especially critical for ADCs, or antibody-drug conjugates.
ADCs are fascinating. They are basically targeted chemotherapy, right?
Yes, they are incredibly complex triple-threat systems. You have an antibody that targets a cancer cell, a highly toxic chemotherapy payload, and a chemical linker holding them together. The goal is for the antibody to find the cancer, get absorbed, and then the linker breaks, releasing the poison only inside the tumor.
But if it breaks early,
If that linker breaks prematurely in the bloodstream, you unleash pure chemotherapy into the entire body, causing catastrophic toxicity. The Kirkovsky approach mandates rigorous clinical bioanalysis using highly sensitive mass spectrometry to track exactly how, where, and when that complex ADC breaks down in a living human. Mastering that bioanalytical data is the only way a company can confidently scale a highly toxic oncology program without accidentally causing widespread collateral damage.
and how do companies manage the sheer volume of data coming from all these AI predictions, ADME profiles, and clinical bioanalysis? That is what Zentalis Pharmaceuticals is tackling with their clinical strategy architecture.
Precisely. You cannot run a billion-dollar phase 3 clinical trial on a hunch. Zentalis utilizes a rigorous clinical strategy architecture built on predictive data integration and highly disciplined program governance. They take the data foresight generated by the AI models, combine it with the early-stage ADME bioanalysis, and construct a rigid mathematical architecture for their late-stage oncology development. It removes human bias and emotional guesswork from the equation. They only advance a drug to massive, expensive late-stage trials when the integrated data architecture provides predictive certainty that it will be both safe and efficacious.
But even if we perfectly design, manufacture, and test these incredible therapeutics, we hit a massive societal wall. Global access and healthcare policy. What good is a mathematically perfect, non-toxic drug if a patient in a developing nation or even a middle-class patient here cannot afford it?
This is where we hit the concept of policy ignition. For decades, policymakers have tried to fix global pharmaceutical access through isolated, fragmented reforms. They might try to forcefully lower prices or alter patent lengths, but these isolated tweaks rarely work and often stifle innovation. Policy ignition is the growing realization among global health economists that access only genuinely improves when intellectual property systems, healthcare payment models, and real-world evidence data streams are structurally engineered as interoperable feedback loops.
Explain what an interoperable feedback loop looks like in drug policy.
It means the systems talk to each other automatically. If a new TPD drug is proving to be highly effective and perfectly safe in real-world clinical evidence, meaning patients aren't just surviving, they're going back to work, that data must immediately and automatically feed back into the payment structures to ensure broader insurance coverage. Simultaneously, the intellectual property frameworks must be legally structured from day one to allow for licensed, scalable manufacturing in developing regions once specific safety and efficacy benchmarks are hit. It cannot be an afterthought. It has to be an integrated global system, exactly like the integrated biological systems we are trying to heal.
That is a massive structural challenge for society, but I want to bring this all the way back down to the individual human level, sitting in a hospital bed. We have an incredible example here of how these predictive algorithms are saving lives right now, today. The sepsis shadow.
This is one of the most powerful real-world applications of everything we have discussed. Sepsis, a life-threatening systemic cascade caused by the body's extreme response to an infection, is a massive, rapid killer in hospitals. By the time a patient shows obvious clinical signs of sepsis, their organs are already failing. The window to save them is closing fast. But researchers are now using machine learning risk mapping to identify hidden, interpretable risk signatures before the patient ever crashes.
And the research specifically focused on stroke patients who subsequently developed bloodstream infections. Why stroke patients?
Because a severe stroke doesn't just damage the brain. It profoundly alters the body's entire physiological and immunological landscape. It causes systemic immune depression, making the patient uniquely vulnerable to hospital-acquired bloodstream infections that rapidly spiral into sepsis. To predict this, researchers utilized regularized lasso models.
Okay, let's explain a lasso model for you listening. LASSO stands for Least Absolute Shrinkage and Selection Operator. It's a type of statistical regression, but what does it actually do?
Imagine you were looking at a patient's electronic health record. You have thousands of overlapping variables: age, blood pressure, heart rate, white blood cell count, exact location of the stroke, medications. It is a sea of noise. A regularized lasso model acts like a mathematical sieve. It analyzes all those thousands of variables, and it actively shrinks the irrelevant ones down to absolute zero. It throws out the noise and leaves you with only the highly specific, tightly correlated variables that actually matter.
It finds the signal in the noise.
Exactly. And the lasso model identified a highly specific, interpretable risk signature from the clinical data of these stroke patients. It essentially created an algorithmic early warning system. It allows intensive care doctors to look at a patient's chart and see that they possess the exact mathematical signature for an impending bloodstream infection days before the severe physical symptoms ever appear. This allows for targeted, physiology-aligned clinical management.
So they treat the future infection.
Yes. The doctors can administer specific prophylactic antibiotics or alter the patient's care based on predictive data. We are fundamentally shifting the paradigm of medicine from reactive care, desperately trying to save a patient already crashing from sepsis, to proactive care, treating the physiological shadow of the infection before it ever materializes.
It's taking the massive computational power we are using to predict HERG cardiotoxicity and applying it directly to a patient's bedside monitor in the ICU. It is truly remarkable. Which leads me to a final provocative thought for you, the listener, to mull over as we conclude. We have explored the terrifying elegance of biological toxicity, the 4 steps of cellular destruction, the blinding complexity of 3D chirality, and the dawn of agentic AI designing self-correcting drugs. If agentic AI and fully autonomous robotic laboratories eventually master 3D chiral intelligence perfectly, if targeted protein degradation allows us to precisely erase any pathogenic molecule without collateral damage, will we reach a point in human history where drug toxicity is no longer considered a tragic biological inevitability, but simply a computational error of the past?
If the AI models reach a state of true predictive perfection, if they can accurately simulate the entirety of human physiology and every possible 3D molecular interaction before a chemical is ever physically synthesized in the lab, then yes, an adverse effect would no longer be an unavoidable, accepted risk of practicing medicine. It would represent a fundamental failure of the algorithm. We are looking toward a horizon where the word side effect might disappear from the medical vocabulary entirely.
Before we wrap up, thank you to all our partners, sponsors, and scholars who made this podcast and the research possible. We're truly grateful for your support. And to our listeners, thank you for being part of this journey with us. We hope you enjoyed today's discussion. If you did, be sure to subscribe and support us in Patreon. And of course, you can write to us in comments if a specific topic needed to be covered, which help us all to grow. Until next time, stay curious, keep learning, and let the search for wisdom guide you forward. Have a great day.